Hi, my name is Mary Mullins, and I'm the program chair of the Developmental Stem Cell and Regenerative Biology program in CAMB. I'm very happy to be able to talk to you today to tell you about the research we do and the activities in DSRB. And I'm going to deconstruct this by telling you about the three areas of research that are focused on in this program. So starting with developmental studies, these focus on cell fate decisions and regulatory circuits that determine these cell fate decisions through cell signaling, transcriptional networks, and epigenetic pathways, as well as single cell methods like single cell RNA-seq, etc., as well as studies of morphogenesis, including growth control and how tissues and cell types are shaped. So the work of Paul Gadou's lab really intersects in multiple ways with the DSRB areas of research, both in terms of stem cells and in development, human development actually. So Paul's lab focuses on using induced pluripotent stem cells to study human diseases. And this is the work of a current student in Paul's lab, Sid Kishore, who discovered from human patient tissue a mutation in the GATA6 gene in the 3' region of the gene that leads to an effect on binding of an important transcriptional regulator, ROR alpha, and this affects expression of the GATA6 gene. And this is like a new mutation in a common agenesis phenotype, where in place of pancreas cells, tissues instead form stomach tissue. And Sid recently published this work in Cell Stem Cell. Work from Montserrat Anguera's lab, and the current student Isabel Sierra in her lab are studying X inactivation, and a long non-coding RNA exists that coats the inactive chromosome, and they've developed some allele-specific oligo paints in a collaborative study with Eric Joyce's lab in the genetics department, and they're using these to understand how the inactive X chromosome is regulated as naive B cells become activated, and they see actually a change in exist localization during these stages. And they're also interested in topological associated domains and megadomains and how particular genes on the X chromosome escape X activation through possibly loop-out domains. Ed Morrissey's lab focuses on lung development as well as lung regeneration and lung stem cells. And shown on this slide is work from a student, Yi Wang, in Ed's lab, where they studied chromatin remodeling and its regulation of microRNAs. Through TGF-beta pathway and extracellular matrix regulation in the morphogenesis of these AT1 alveolar cells and how they become spread thin through this process, and this allows the gas exchange. So this is some work from Yi Wang that was published in Developmental Cell. Moving to the second area of research, stem cells, which are really key cell types in renewing organ systems as well as in repairing tissue after injury, and studies include the stem cells themselves as well as their niches, stem cell behavior, and their differentiation during these processes. So the work from Nancy Speck's lab has focused on hematopoietic stem cells, and they discovered a really important transcription factor, RUNX1, that is key to the formation of these hematopoietic stem cells in the mouse. This is work from Amanda Yazguir, a former student, now a postdoc at the Salk Institute. And some follow-up studies from this are using single-cell RNA-seq to understand how RUNX1 is regulating and allowing these cells to turn into hematopoietic stem cells. Turning to the last area of research, regenerative biology, where tissues are repaired, and this can occur in normal physiology or in disease states, or following injury. And of course, iPS cells are an important part of this research as they're used to intervene and manipulating them to repair tissues in restorative medicine. The work of Chris Legner's lab highlights intestinal stem cells that are key following DNA damage through a Musashi mTORC axis downstream of nutrient sensing that has shown that Musashi is both necessary and sufficient to drive exit from quiescence and entry into the cell cycle in this regenerative process. And this is work from Mariam Yousefi, who is currently a postdoc now at Stanford. And then there's the work from Michael Granato's lab and his student Allison Rosenberg. And they're using the zebrafish to study regeneration and degeneration of motor neurons. And they use, the zebrafish is translucent, so you can use these fluorescent reporters to analyze the degeneration process, which is shown here in this time-lapse movie, and also regeneration, and determine the molecular cues that are important in these processes. So in addition to the great research going on, we also have important training in our program that focuses around rigorous scientific methodologies, using cutting-edge approaches and technology. We focus on training in presentation skills, scientific writing, with an emphasis on professionalism, and there's lots of collaboration that goes on within this program. There's lots of advising at all steps of your graduate career. Mira Sundaram and I are the first-year advisors. Patrick Seal, Wenchen Liu, Paul Gadou, and Fanchen Long have been the second-year advisors. And Faye Morchiotti and Dan Kessler provide advising on presentation skills. We have bi-weekly DSRB lunches, which are research in progress talks, a journal club that's run by the students over pizza and beer twice a month. And we also have student-invited seminars, 2 to 3 per year, DSRB peer mentoring, as well as superb prelim advising. We have great success in NIH fellowships in DSRB with a 70% success rate. We do lots of fun things. We have social hours, an annual DSRB retreat, Quizzo nights, and here is at our DSRB retreat, a PPE contest that we had lots of fun with. So this is a video that was made by our students showing Dan Kessler here and Steve DiNardo, the former DSRB chair, for one of the Quizzo nights. So if you have any questions, feel free to contact me at my email address, and I look forward to you being here at Penn in the